Welcome to week 11 and this talk on protein structure. This is the second part. And what I'd like to talk about during this podcast is a little bit about structure, protein structure prediction, and a little bit about protein databases. Let's start with protein structure databases. As you can see from this slide, databases of three-dimensional structures of proteins have been calculated mostly using either a technique known as X-ray crystallography or what's known as nuclear magnetic resonance or NMR techniques. So basically what has happened over the past couple years is that scientists have been able to elucidate or discover the atom by atom structure of many proteins, and these structures have been uploaded to a wide variety of databases from which you can do protein studies. And four of the more famous protein databases that we're going to use, we'll use two or three, maybe even four of these databases in the next couple weeks. Certainly the most well-known one that you've already had a little bit of experience with is the Protein Data Bank, or PDB. We also, I think, introduced you a little bit in the past session to the SwissProt database. There's also the PIR, the Protein Information Resource. PIR is particularly nice because it's a little less technical in some regards than PDB and SwissProt. So PIR is a nice place to go for beginning studies. And then the fourth database is the SCOPE database, the structural classification of proteins. So in the Illuminate session, the live session, will demonstrate and talk about a couple of these protein databases so that you can use these for continuing studies. You should know by now that there are four levels of protein structure. The primary structure is just the amino acid linear sequence, and we had you look at hemoglobin with its 147 amino acids. And then what happens is these linear sequences fold into the first step is sort of a secondary structure, which consists of either alpha helices, beta sheets, and loops or coils. And then that secondary structure folds one more time into a tertiary structure, the 3D shape of the polypeptide or polyamino acid chain. And then there's finally a quaternary arrangement, which is just an arrangement of several polypeptide chains. So one of the things that you're trying to do in protein structure elucidation is really try to get a sense of the four levels of protein structure and being able to break a protein down into its four levels. Secondary structure, quick reminder that usually divided into mostly two, sometimes three categories. We have the alpha helix, we have the beta strand or the beta sheet, and anything else that's out there is a turn or a loop. So those are the secondary structures. Okay, and let's now talk a little bit more about the Protein Data Bank, or PDB. It holds 3D models of biological macromolecules, proteins, RNA, DNA. And one of the great things about it is all the data is available to the public. It's not proprietary. You don't have to pay money for it. Most of the data there has been obtained by X-ray crystallography. This is, by the way, the technique that Rosie Franklin used to help Watson and Crick figure out the structure of the DNA molecule. So most of it is X-ray crystallography. Some of it is by NMR spectroscopy. And anybody can submit to the PDB, biologists, biochemists from around the world. So if you happen to elucidate a protein structure, you can upload your structure to the PDB, and everybody will be very appreciative. The PDB was founded in 1907, so it's been around a little while at the Brookhaven National Lab in New York, and it was transferred to the Research Collaboratory for Structural Bioinformatics, or RCSB, in the late 90s. And you see the crossed out thing here. Currently it holds 55,271 released structures. I think this is 2005 data that I have on here. So this number changes literally every day. I mean, there's proteins being uploaded literally every day. So this number is just changing constantly. Okay, a little bit about a PD model. It holds the 3D positions of atoms in one or more molecules. And these can be proteins, protein complexes, proteins in DNA, protein segments, Sometimes they'll also include the position of ligand molecules, solvent molecules like water, metal ions like in hemoglobin has a iron in there. Okay, all kinds of things can be in there. And you have to be a little careful when you're looking at PDB structures because sometimes you'll have a protein without a ligand, sometimes you'll have a protein with a ligand, sometimes you'll have the protein without water, sometimes you'll have it with water. So you have to just can't blindly pick one of the PDB uh, molecules and think that that's the right protein because there's lots of variations on a theme. Okay, the PDB file, the text format, a little bit about this. These are all text files, and you should be able to see these pretty easily. Excuse me 1 minute. Pardon me, thank you. Okay, so PDB file text format. Um, These are all text files, and you can look at this, and you should probably try to open up one of your PDB files in Notepad or some other text format, so you can get a sense of what this data looks like. So, real quickly, you see it has the atom identity, it has what residue it's in, it has what chain it's in, it has the XYZ coordinates for each residue in the structure. So. You see down there where it says het atom, that's usually a ligand or like an ion or a water. So it's important that you have some sense of PDB structure. So take a look at one of those with your text editor, notepad, whatever the case may be. Okay, here's just a picture of the protein data bank. And again, this is linked on the MedChem page. We will be going to this fairly often. Okay, how many structures? Again, okay, this is 2005 data, and you can see the chart at the bottom is probably the easiest thing to see, that the number of proteins being added to the data bank is exponential. Okay, how do you see these proteins? There's a number of programs out there that convert these atomic coordinates into 3D structures so you can view the molecule. It allows the user to manipulate the molecule. You can rotate it, you can spin it, you can zoom it. And this is really critical in drug design because this is going to yield some insight into how the protein might interact with ligands at an active site. Some of the more popular programs for viewing 3D structures, absolutely my favorite is Protein Explorer, which you can download and install on your machine. I think Rasmol is still out there, Chime, CN3D, Kinemage out of Duke University is a good one. SwissProt has a viewer called Swiss 3D Viewer. And if you go to the Protein Data Bank page, you'll see a whole list of viewers there. There's King, there's WebMol, there's JMol, there's a whole bunch of viewers. And pretty much you have to figure out which viewer you like the best. Again, my favorite is Protein Explorer because I can click on individual amino acids and see where they are in the protein. And but again, you'll figure out which one is your favorite. Okay. What we're going to want to do, and I'm going to breeze through this pretty quickly, is eventually we're going to want to be able to do some alignments of protein structures, being able to compare a 3D structure of one protein against a 3D structure of a second protein. And we're going to be doing this to help us look at similarity patterns to be able to predict what this protein might be doing and how it may be reacting in the body. Okay, so here's an example where I've taken two proteins and I've done a little bit of alignment. This is a little bit of introduction, by the way, to what we'll do next week when we talk about bioinformatics. So we're giving you a little taste of where we're going with this. And this is why you need to understand protein structure. To be able to align these things, you have to understand sort of a little bit about what you're trying to look at. Okay, so real quickly, I'm going to breeze through these. Okay, here's two cartoons of proteins. And I can do what's called a transformation, and I can superimpose to see how well these proteins line up. And by lining these proteins up again, what I can do is I can make some statements about the protein structures and how they may interact, and some things that they might do in terms of biological activity. Okay, so we'll do a little bit more on that, and you'll do some alignments next week. Let's talk a little bit about protein secondary structure prediction. And why do we need to do this? We need to do this because accurate secondary structure prediction is really important for being able to figure out the next levels, tertiary, quaternary, it's also really important for figuring out what the protein might do. It's useful for figuring out protein classification. And it's also, we need to be able to do this so we can predict what structural changes might happen, especially when we stick a drug in there and get it to do something. And we've been trying to predict protein structure for a long time, over 40 years now. Um, and we're getting better at it, but we're still not there yet. Okay, so real briefly, uh, you should know something about secondary structures here, and there's what the sort of the classic toilet roll representation of main chain hydrogen bonding and an alpha helix. By the way, you should notice that there's an amino end and there's a carboxy end to these structures. About 30 to 35% of proteins are alpha helices, and they're typically right-handed turns. These are the most abundant secondary structure, and so these are, and you notice the um, amino acids there, alanine, cysteine, leucine, methionine. These are the ones that are the primary alpha helix formers, okay? So those would be good to know. You should also know, by the way, you should have a little cheat sheet that you keep with you of the one-letter codes for all 20 of the amino acids. So I would have that at your disposal all the time. Okay, beta sheets comprise about 20 to 25% of the secondary structures. And again, these are hydrogen bonds between groups across strands. And these are a little bit less compact. And again, there you see where beta sheet formers are V, I, P, T, and W. And I leave it to you to figure out which one these are. You also have beta turns, which is a little bit different than beta sheets. These are short turns, usually 4 residues, and they're very polar and oftentimes found near the surface of the protein, and that's going to be important to know. And beta turn formers are SD, N, P, and R, and again, I leave it to you to figure out which one of those are. So you might want to consider classifying your amino acids by whether they form alpha helices, beta sheets, and beta turns. That would be a good activity for you to consider doing. And there's some others out there. There's coils, there's turns, so loops and coils. So these are regions between alpha helices and beta sheets. They don't have any regular periodic structure, and loops are typically small polar residues. And a coil, which is the rest of the protein, is anything besides an alpha helix, a beta sheet, or a beta turn. Okay. So loops and coils are going to have some differences and similarities, but basically we classify these as sort of other secondary structures. Okay, assigning secondary structures, there's a variety of ways to do that. We can define the dihedral angle, the hydrogen bonds, and the geometry. Those of you who took computational chemistry should have some familiarity with these issues right here. We can assign these manually by crystallographers, so people who do X-ray crystallography do this. There's also a number of algorithms, for example, the DSSP, stride, and continuum, a variety of algorithms by which, or mathematical calculations by which we can define these secondary structures. And I'm just going to flash this slide up here, but there are a variety of codes that we use. So for example, you would use an H for an alpha helix. There's different codes that we would use for indicating the secondary structures of proteins. There's the CASP standard, okay, and we'll show you some pictures of these. We'll have you do some of these during the lab. Okay. So for example, here we give you a protein structure, and we just gave you a short snippet there of amino acids. So this is probably 20, 25 amino acids or so. And what we would like you to do is predict its secondary structure content. Coils is C, H is alpha helix, E is beta strands. And so there we've applied a mathematical method, and what we're predicting is that secondary structure, where again, E is the beta sheet, H is the alpha helix, and C would be the coils. Okay. There's a number of algorithms. There's probably a dozen or so by now. And for this slide, I'm going to show you the Chou-Fasman method, which is probably the simplest method there is, and we'll go through it very briefly. So what we want to do here is examine 5 to 6 residues. Residues, by the way, is another name for amino acids, to predict the structure. And from a PDB database, what we're basically trying to do is calculate a value called the propensity for a given amino acid to adopt a certain secondary structure type. And so we use this algorithm here to be able to predict that. So we gave you a sort of an example there. So predicting the propensity for alanine to form an alpha helix, I gave you, or we give you some rough numbers there in a particular protein uh, um, of 20,000 amino acids. 2,000 of them might be alanine. Uh, there are 4,000 helices. In those helices, say there's 500 alanines in that helix. What's the p-value for that? And we can calculate that as being about 1.25. That number is actually, we gave you whole numbers there, so you can do the math sort of in your head. Okay. Turns out that the propensity value for alanine, if you look at this chart here, so the propensity of alanine to form an amino, or excuse me, an alpha helix is about 1.42. The other way, most times people will report those. So that's basically a percentage. So basically there's 140, there's 142% that thing might form as an alanine might form into an alpha helix. So typically we report these things as a percentage. So instead of the 1.42 there, you think of that as 142. Okay, so the way the Chou-Fasman algorithm goes, I'll leave this up here for a minute so you can sort of glance through it. We scan through the peptides, identify regions where 4 out of 6 contiguous or in a row residues have a propensity greater than 100, and that's called an alpha helix. And then we extend that helix in both directions until the set of 4 continuous residues has an average of less than 100 is reached, and that's the end of the helix. And if the segment defined by this procedure is longer than 5 residues, okay, that can be signed as the helix, and we repeat this for all the things. You can see it's very complicated mathematical technique, but if we give you an example here, so we gave you, I'm giving you a short segment here, and what we want to do, say, let's look at that 6 residue segment there, TSP-TAE, and I'm looking for where the pH is greater than 100. Okay, are 4 out of 6 of those greater than 100? The answer is no. Okay, so I move over and look at the next contiguous group. So there's the PTAELM. Are 4 out of 6 of those greater than 100? The answer is yes. Okay, so that's going to be partly where my alpha helix is going to be. Okay, now I extend that helix in both directions until a set of residues has, let's go in 4. Okay, there's 4. Okay, until that has an average pH of less than 100. Okay, I go 4 this way. Okay, and 4 that way, and I keep going 4. Okay, until I get a value, okay, that is going to be less than 100. So the pH there is 107.5%, and that's greater than the PE, which is 85%. So that area there is assigned as a helix. Okay, so and then my prediction is going to be that all of that is a helix right there. Okay, the beta strand, basically we're going to use a similar algorithm. Okay, we look at the PE. Okay, and so there's a different algorithm for that. And again, so if I want to predict here's an exercise. If I want to predict the secondary structure, this is the mathematics that would need to be done. Okay, so based on these numbers here, I'm able to predict where the alpha helix is. That's the H value. I'm able to predict where the um, um, beta strains are. That's the E value. I'm able to predict where the turns are. That's the T value. And so at the very bottom of that exercise there, I have my predicted secondary structure for this molecule. Okay. There's lots of different ways to do this. We can, lots of prediction methods. We can examine a single protein sequence. We can use statistics. We can use something called neural networks. We can do alignments, as I talked about a little bit earlier in this presentation. And there's lots of different ways out there to be able to do this. Um, all of them have differences in terms of accuracy. The Chou-Fasman method I showed you here, I picked that because it's pretty simple, and it's still hard, but it's simpler than the rest. Has about a 50% accuracy range. Garnier has a little bit higher. Uh, Zevabil, Lee, and Landers are a little bit higher, and some of the neural network methods are starting to approach the 70, 75, 80% range, so they're getting a little bit more accurate. All these methods are getting better as we get smarter about the mathematics and the chemistry. Okay, so I did a what's called a Pele prediction on the student biology workbench, and this is one of the things that I'll ask you to do for your protein evaluation. So I chose myoglobin from the human. This is muscle protein. And so you see, I only showed you part of the sequence. I didn't show you all 154 amino acids, but I showed you most of them. And so what you see here is the sequence in black along the top, and we've numbered them to make it easier. And then below that, I have 1, 2, 3, 4, 5, 6, 7, 8 different protein prediction methods that are applied to this amino acid. And what you want to look at here is see where these protein prediction methods either agree or disagree. So say looking from residue 40 to 50, okay, it looks like almost everybody predicts that that's with possible exception of KS. Most folks are predicting between 40 and 50 there at the top right, that those are probably going to be alpha helices. Okay, the KS method thinks that 40 and 41 are not yet in the alpha helix. So we're going to ask you to look at that during the lab. At the University of Virginia, they have a protein prediction site. Actually, there's a ton of these on the web. So I took my myoglobin primary sequence, and I chose the Chou-Fasman prediction method. I entered it as FASTA format. I hit the submit sequence button, and basically this is what I got for my Chou-Fasman. So you can see where there are turns, where there's beta sheets. There's a couple places there where nothing is printed, so that's going to be a coil. A couple places where it's not real sure what it's seeing there. So this is what the Chou-Fasman method gives me at the University of Virginia site. And that's the end of this presentation. We'll see you online. Thank you very much.